Good afternoon, everybody. My name is Dr. Anita Go. I'm a clinical neuropsychologist and a senior research fellow at the National Ageing Research Institute. And I'm also the founder and immediate past chair of the Australian National Younger-onset Dementia Special Interest Group. So welcome to our online seminars in ageing program for today. I'd like to begin by acknowledging the traditional owners of the land on which each of us are meeting today and pay our respects to our elders past and present. For me, it's the Wurundjeri, Woiwurrung and Bunurong people on whose country I am now. And Aboriginal and Torres Strait Islander peoples from Australia are the first scientists of this land, and we should be committed to preserving that history and learning from their knowledge. So firstly, a little housekeeping regarding Zoom today. At the conclusion of the talk, we'll have some time for some Q&A, which is always a great part of these seminars. So if you have any questions, you can submit them throughout the seminar using the chat function, which can be opened using the chat button at the bottom of your screen. I'll be keeping an eye on that so I won't forget your questions for Olivier. If you do have any really burning questions, feel free to pop them in and let me know if they're urgent, for example. So today I have the great pleasure of introducing our speaker. So today we'll be hearing from Professor Olivier Piget. Professor Olivier is a professor of clinical neuropsychology and NHMIC Leadership Fellow at the University of Sydney, where he's the director of Frontier, the frontotemporal dementia clinical research group at the Brain and Mind Centre in Sydney, Australia. He's a clinical neuropsychologist with over 20 years clinical experience in the field of FTD and related conditions. Olivier trained in Geneva and Melbourne and completed his PhD at the University of Sydney, followed by a postdoctoral fellowship at MIT. His research program investigates early clinical markers of frontotemporal dementia, prediction of disease progression, and relations to biomarkers, genetic, and brain pathology. He's published over 300 peer-reviewed journal articles on these topics, attracting over 19,000 citations. Olivier is a founding member of the International Society for Frontotemporal Dementias and current president of ASBE, which is the Australian Society for the Study of Brain Impairment. So today we're very lucky he's presenting things we know, things we wish we knew, challenges in diagnosing frontotemporal dementia. So welcome, Olivier, and over to you.
Thanks, Anita, and welcome everybody. I'm on Gadigal land in Sydney here, so welcome here and welcome wherever you are. I'm going to share my screen. All right, hopefully you should see the slides. Yes, can I have a yes from the audience? Yes, excellent. Thank you, thank you. Yes, I've been working on FTD for some years now and listening to this introduction, very kind introduction. Well, it's the bio that I submitted, but it's interesting to have it read by someone else. But I realized that I'm still working on the same things I've been working on for some years, which are issues of diagnosis, prognosis, and trying to understand clinical pathological correlations in these disorders. So things we know, things we wish we knew, and it's still the same issues. And it goes back to Donald Rumsfeld's famous quotes. There are known knowns, there are things we know. And we also know there are knowns, unknown unknowns. That is to say, we know there are things we do not know. But there are also unknowns, the ones we don't know. So I'm going to talk about known knowns and some known unknowns in the field of FTD. And maybe that will lead to trying to identify some of the unknown unknowns that we need to tackle in the future. If you're affiliated with NARI, there are things that I'm going to talk about. I'm going to go over these things very quickly because you know them. So these are the known. And the first one is, in Dementia in Australia, we're talking about close to 500,000 people with dementia today, and which means 200 new cases per day. And when we break that down, it comes down to about one person every 6 to 7 minutes, which is really scary. And then if we do nothing by 2051, we're going to see an increase, about threefold increase in the number of cases of people with dementia, which translates into 650 new cases a day, or about one person every 2 to 3 minutes. Dementia, as you know, is the leading cause of death in women and second leading cause of death overall. Again, which is quite scary when you think that with the aging of population, this is an immense problem, health problem that we need to tackle, particularly in the context of the lack of disease-modifying treatments. Now, I always like I don't like this figure, but it's an important figure. So $15 billion, that's the cost of dementia in Australia, which is 1% of GDP. Again, if you break that down, you can get to staggering figures. So it's $1.8 million per hour. So by the time I finish this talk, about that amount of money will have been spent, or that amount of money will, dementia will have cost that amount of money. And again, if we do nothing, we're looking at $36 billion in terms of direct and indirect costs related to dementia. So in terms of the frontotemporal dementia, and I was not sure how much you would know about the background of these young onset dementias. So I've just put a few background information there. And that's what I've called the known number 2, and ish, because we know some of these things, but it's still not very clear. So when it comes to frontotemporal dementia, what we're talking about is really dementia syndromes that are mostly common occurring before the age of 65 years, so younger onset, and with a typical curve of incidence that is at the highest between the age of 50 and 70. We do see patients in their 20s, rarely, but we do see them, and the 30s, 40s, yes. We also see people with confirmed frontotemporal dementia in their late 70s and early 80s. So it's not just a younger onset dementia syndromes, and that's something probably to keep in mind. Just because you're old doesn't mean that you can't have frontotemporal dementia. Then in terms of survival, it's approximately 8 years from symptom onset, but I'll come back to that in a minute because it's highly variable. And there are some factors that people can keep in mind to that will inform the likelihood of prognosis or disease duration in the patients that they see. In Australia, we estimate that, out of, I think about 40,000 people with young onset dementia, probably about 20,000 of these will have FTD. It's really hard to know the actual numbers because of the confusion of diagnosis with other disorders. When you get diagnosed, when you present with behavioral changes or cognitive changes, when you're in your 40s or 50s, dementia is not necessarily the first diagnosis that comes to mind, and so confusion with other disorders like primary psychiatric disorders is very common. So it's an estimation 20,000, probably more. And the estimated costs, again, which is interesting. So it's estimated that it costs in a direct and indirect cost and loss of earning about $125,000 per person per year, which is twice as much as someone diagnosed with Alzheimer's disease. So in terms of the main presentations of frontotemporal dementia, we're dealing with 3 different syndromes, and that's part of the source of the difficulty in trying to understand these disorders. Unlike Alzheimer's disease, the presentation of typical Alzheimer's disease is fairly uniform. You know that if you have a cluster of language difficulties and episodic memory loss, the likelihood of having Alzheimer's disease is pretty high. In frontotemporal dementia, we're dealing with 2 main groups. One that is this behavioral variant FTD, and 2 language presentations or language variants. And I'll just go over these very quickly. So in terms of the behavioral variant FTD, as the name indicates, it's primarily a syndrome of change in that outlines changes in social conduct, personality, and you have this loss of empathy, difficulty relating to other people. You have this apathy, loss of insight, and sometimes you have a cluster of things like collectionism, so start collecting different objects. A bit similar to OCD, but without the anxiety associated with it. In terms of cognitive deficits, you see changes in social cognition, so emotion recognition, that is related to the lack of empathy, but also in executive function, and some changes in attention, working memory as well. Then you have these two language variants. The first one is semantic dementia. Also forgot to say that, on this slide, so the behavioral-oriented FTD, on neuroimaging, what you will find is primarily brain atrophy that involves the orbitofrontal cortices bilaterally, and also the dorsolateral and medial prefrontal cortex. In contrast with semantic dementia, you have on neuroimaging a very focal anterior temporal lobe atrophy, primarily on the left, in left hemisphere. And clinically, these patients would present with a very different presentations, where the main complaint is one of loss of memory for words. So these patients will tell you, I can hear words, but I don't understand what these words mean. And what you see is really a broad loss of semantic knowledge. So it goes beyond language, because you can test these people using not only words, but you can use sounds, you can use images, you can use pictures, faces, and so on. And there's this really pervasive loss of meanings of things in the world. So that affects all domains, and it's impaired recognition of objects, faces, and also that impacts on aspects of social cognition. So it's really broader than a language disorder. Then you have also progressive non-fluent aphasia, which is a little bit the other side of the coin of semantic dementia. So semantic dementia is a fluent language disorder. So that when you listen to these patients, the speech is fluent, but it's empty and lacking in specificity. In contrast, the progressive non-fluent aphasia, these patients have an expressive language deficits, where the speech output is really labored, distorted, and effortful. And what you see is a markedly reduced speech output. So these patients also have insight, so they tend to minimize the occasions of interactions because of their difficulties. And when you listen to their speech, what you notice is that the sounds that they use is also incorrect. So it's this distorted speech in that the phonemes that are used, that are put together to make the words, are actually the motor programming of these sounds are incorrect. So it means that the sounds that you hear are really are sounds that don't belong to the language in which they speak. But the comprehension is generally preserved. So so you have these nice contrasts, if you wish, between these two language variants. Now, what we know about these syndromes as well is that there's multiple pathologies associated with frontotemporal dementias. And again, if you compare that to Alzheimer's disease, we know that everyone with Alzheimer's disease will have a deposition of A, beta, and tau in the brain. Whereas in frontotemporal dementia, there are also two proteins involved, but you will find cases with one or the other pathologies. And so you have, as in Alzheimer's disease, the tau protein that is involved, so accumulation of within neurons of abnormal accumulation of the tau protein, and this type accounts for about 40% of all cases of FTD. Then the large majority of cases that do not have tau will have TDP43, so tau DNA binding protein. That can take various forms, but I don't want to go into too much detail here. Then the remaining cases, you can find cases with what has been called fused in sarcoma protein, so fuzz. But then we'll still have about 5 to 8% of cases that don't have something else. They don't have tau, they don't have DNA, the TDP, and they don't have fuzz. So then the challenge is also to understand who's got what when. So when you have a patient coming to your clinic and you need to be able to see if you can determine what's the underlying pathology, because that will have implication in terms of disease-modifying treatments when they are available, which is still not the case. With regard to genetics, I think what's important to highlight is that most cases of FTD are still sporadic, so we don't know the causes for these disorders. However, in about 30%, 10 to 30%, there's a strong family history. So there's a clear transmission from generation to generation where the disease will occur. But the challenge is also that it can take different form across generations. So sometimes to identify a family history in some families is quite difficult. Within these families, so the 30% of patients where there's strong family history, about 80% are caused by these 3 genes, so MAPTau, granulin, and C9orf72. Then you have other rare genes that have been discovered in recent years. But more importantly, there's also about 30% of patients that have a strong family history, but there's no single gene identified. So which really points to the genetic contribution to these disorders in a way that is not a single gene effect. So there's probably a combination of genes, gene profile, and environmental variables that contribute to the emergence of FTD in some families, and in a way that we still don't understand. So it's starting to date a little bit, this review paper, this nice summary of where you find which pathology. So you can see that, TDP43 pathology is found across various phenotypes that go across the many different subtypes of FTD and even beyond, and the same thing for tau, and they overlap on some of these syndromes. And in all these types of pathologies, some are sporadic, so no cause identified, and some are familial, so there are some genes identified. In fuzz, so this small proportion of patients with the underlying fused and sarcoma pathology, they're all sporadic, so there are no genes associated with the emergence of this type of FTD. So, okay, I've spoken a little bit about these known knowns. Now I just wanted to focus on a couple of known unknowns. And so the things we know, we don't know, or we don't know very well. The first one is really about diagnostic accuracy and clinical variability. The history of FTD is quite long, and the first cases of FTD were described in the late 19th century by Arnold Pick, who was a Czech neurologist. And he described some case series in the literature in the late 1890s and early 1900s of these focal brain atrophy associated with language disturbances. But as recently as 1967, and even more recently, what we have in the literature is a description of pre-senile dementias, and I've highlighted the part here that is important. The most common of these disorders is Alzheimer's disease, which is definitely a clinical pathological entity, no matter at what age, blah, blah, blah. Pick's disease, which with a similar clinical picture and age distribution is much less common. So at that point in time, there was a belief that Pick's disease or frontotemporal dementia was presenting in the same way as Alzheimer's disease and with the same age distribution. Then more recently, in the late 90s, there was this work led by a group in Lund in Sweden, in Manchester in the UK, that were focused on these younger onset dementias and identified tests that could differentiate clinically patients with Alzheimer's disease and patients with underlying frontotemporal dementia. But then there's the attempt of a first consensus diagnostic criteria paper. But even in the early 2000s, there's still papers that are being published where there's this paper by a group in Boston where they outline the fact that it's very difficult to differentiate Pick's disease, which is a form of FTD, and Alzheimer's disease on clinical findings and rate of cognitive decline. And again, the same work being done trying to identify markers of the disease that would be specific to FTD. And it's only in 2011 that we have these two seminal papers, one published in Brain, looking at the behavioral variant frontotemporal dementia, and the other one published in Neurology the same year, looking at the language presentations called the primary progressive aphasias. And where they identified core features of these syndromes that would help with the classification and also then treatment and management of these patients. So the difficulty here is that, as I said, we have these three syndromes. They're also related to other motor disorders like MND on the one side and corticobasal degeneration and progressive supranuclear palsy. So what we see over time is that rather than having these nice classification, these nice grouping of patients, is a situation like this one where you have patients who will present with multiple shades of different things. So you can be presenting with behavioral brain FTD and over time develop signs of motor neuron disease, or vice versa. You can present with progressive non-phenophasia, but over time you can also show signs of behavioral changes or motor symptoms associated with corticobasal syndrome or progressive supranuclear palsy. So essentially, it's a bit of a mess in terms of diagnosis and how to really find markers of disease that are specific to one and not just sensitive to multiple syndromes is really difficult. Now, I've mentioned, which is the second known unknowns, in clinical practice, when you see patients, there are three things they want to know is, well, what's the diagnosis? Then once you've then a little bit and say, well, I think it looks like it's more likely to be, behavioral variant or semantic dementia or whatever. Then the next question is, well, what's going to happen next? And often it's a difficult conversation to have because at the broad population level, we know what's happening, but there's no way to predict disease progression at an individual level yet. And then the third question is about treatment, which again, we don't know much about. So as I said, the survival from symptom onset or from time of diagnosis is highly variable. And if you have, what we know is that if you have a coexisting motor neuron disease, this is really bad news because on average, the disease duration is about 2 years and ranges between one and a half years to 4 or 5 years. If you have a behavioral presentation, it's slightly longer, but again, we've people in our clinic who've presented and were dead within 2 years, and we still have cases we started here in Sydney in 2007 and are still with us. So 15 years down the track, where some of these patients are still with us and they are progressing, but at a much slower rate. And how to understand these differences is really critical for disease management and also for informing families. Then if you have the language presentations, it tends to be longer. And particularly if you have semantic dementia, they have much longer disease durations. So what we see also at an individual level is that there's really large inter and intra-individual variability, and that's work that's been done by David Fox, who completed his PhD earlier this year with us. And you can take any measures you want at a clinical level. He will look at mood in that particular study. And you can see that the profiles over time are highly variable. You have the more typical expected progression we'll take this one, where over time there's an increased mood disorder that tends to occur in these non-fluent patients. It was focusing on the progressive non-fluent aphasia. But you also have patients who tend to improve over time, and you have some that just go all over the shop, or some that don't change at all. So again, trying to understand what is happening there at an individual level is really important. So then the third known unknown is about prediction of pathology. I've hinted to that a little bit earlier, where we have different pathologies. And so we have our tau and TDP. And here's just to show you the relationship between the syndromic presentation and the different pathologies. And in each broad type of pathologies, so tau, TDP, and fuzz, you have multiple subtypes. So you don't have just one form of tau, you have at least 4, and the same for TDP, and the same for fuzz. So if you take the language presentation, it's fairly clear and fairly easy to ascertain. So if you have a diagnosis of semantic dementia, in a large majority of cases, you will have TDP-43 type C. In a few cases, you will end up with Pick's disease, so a tau pathology. And the same thing with the progressive non-fluent aphasia, which tend to be related to underlying tau pathology. Then if you look at the motor syndromes, so MND is associated with TDP and fuzz, but not tau, which is still fairly circumscribed. And the other motor syndromes affiliated or associated with FTD, the CBS, corticobasal syndrome, and PSP, are more affiliated with tau pathology. Now, the last one is the behavioral variant FTD, which is the most common subtype. And here you might as well toss a coin, because it can be associated with anything, essentially. So when you put all these together, again, it's a big mess. So essentially what it's telling us is, unless you have semantic dementia or MND, essentially it's really difficult to predict any of the underlying pathology at a clinical level. And that was a very recent paper that was published, I think, that outlined this complexity in Frontiers Neurology. So just looking at the relationship between, on the one hand, the clinical presentations of various syndromes associated with tau pathology in the brain. And you can see here the pathology. So if you follow one, for example, amnestic presentation, the majority are related to underlying Alzheimer's pathology, but it can also be associated with tau pathology of various forms, CVD, Pick's disease, and so on. So the work that we've been trying to do is to see how we can improve that. And that's work that I've been doing with Ramon Lendin Romero, the patients that we see will have an MRI scan. We see them longitudinally, so on a yearly basis. We have MRI scan on multiple time points, and just to see how we can differentiate these groups of patients over time. And at baseline, we have two very different presentations that have been reported before. So in yellow, you have Alzheimer's disease, which is more posterior, cortical atrophy involving the temporal parietal areas, as well as a posterior cingulate, and also involving the hippocampus, and in blue, the behavioral variant FTD. So this orbitofrontal and anterior medial frontal atrophy. And then if you look at the progression of disease over time in Alzheimer's disease, it tends to move more anteriorly, both laterally and medially. Whereas in behavioral variant FTD, you tend to have a progression that moves posteriorly. So it will tell you something. This longitudinal imaging will tell you something if you can define or identify a pattern of progression of disease, even if you don't know the clinical syndromes and the pathology, based on the progression of where the brain atrophy is, will give you some clues as to the underlying pathology. So if you have something that moves more posteriorly, you're more likely to deal with a disease within the FTD, FTLD space. Then we also looked at doing the same thing where we flipped that over. We took cases where we knew the pathology, so tau pathology versus TDP, and asked the question, well, do they show different patterns of brain atrophy at presentation and over time? And we did find some differences, and where the pathology in the cases with TDP-43 tend to be in blue here, tend to be more posterior compared to the cases with tau pathology, which tends more anteriorly. I don't want to spend too much time on this. It's just to say that from a structural neuroimaging side of things, we can certainly improve the diagnostic accuracy of patients where there's a question around the diagnosis of FTD. We can also certainly map the progression of disease much better now than we used to, and also in terms of clues as to the likelihood of underlying pathological process that is happening in these patients. In terms of pathology prediction, our work seems to indicate that it is possible, but it's still very difficult because we're talking about very circumscribed regions. There's also work that has been done around functional neuroimaging, and as I'm sure you're aware of PET scans that we know that's telling us about metabolism in the brain, and certainly there are some differential patterns of changes of hypermetabolism in Alzheimer's disease compared to FTD, where you have a more anterior loss of metabolism or reduced metabolism compared to Alzheimer's disease, which tend to be more posterior. When we look at molecular PET, A-beta is not really useful in FTD. The problem with A-beta is that also the deposition of A-beta in the brain increases with age. So at some point, you will find A-beta in the brain of people with FTD. But it's probably more age-related than pathology-related. There have been some development around tau ligand that would be specific to FTD, but when in this paper they tested flow tau CP in semantic dementia. So I don't know if you remember, I went through that fairly quickly, but semantic dementia patients don't have tau pathology in the brain. They tend to have TDP. And when you see that they have a high uptake of this tau ligand in the left anterior temporal region. So this ligand tends seems to be related to more neuronal abnormality than tau deposition. Having said that, there are some more recent tau PET, like this 18F MK6240, where there's greater specificity, where they found deposition or, binding of this ligand in patients with a tau mutation with frontotemporal dementia. And same thing, but more variable in pre-symptomatic mutation carriers. There's a bit of binding, and in contrast, in symptomatic non-tau mutation carriers. So people have other types of genetic mutations. So again, in terms of neuroimaging, yes, there's some progress. Diagnosis, FTG PET, yes. Progression, probably not. And pathology prediction in terms of molecular PET, there's certainly some improvement, but not quite there yet. The other useful biomarkers are NFL. And what is interesting in NFL is that it's really related to the emergence of clinical symptoms. So even in pre-symptomatic mutant carriers, they are here in this box. So you have controls, and next to it are the pre-symptomatic mutation carriers. The level of NFL is not different between these two groups compared to the clinical groups where there's clinical diagnosis of these different syndromes, so behavioral variant FTD and the non-fluent and semantic dementia patients. So it seems to be really related to the emergence of clinical features or clinical symptomatology in these patients. It seems to be a marker of clinical emergence of symptoms within a year. So if you take the mutation carriers that did not convert within a year here in the middle, compared to control, it's the same level of NFL, compared to the cases with mutations who will convert within a year, where the overall level of NFL is elevated. The difficulty with NFL is the large inter-individual spread. There's a large overlap. So there's, again, in terms of specificity there's some question mark. Then it's the same with GFAP, which is another marker of neurodegeneration related to neuroinflammation, where we see an increase in various dementia syndromes, not just in FTD compared to controls. But again, you can see that there's a large overlap with the amount of markers that you find in controls. What is interesting with GFAP is that there's an association with cognition, but only in Alzheimer's disease, not in frontotemporal dementia. So regardless of the level of GFAP that you find in the brain, there's a linear association, well, a bimodal association in Alzheimer's disease, where there's a marked increase early on in the disease process, and then it tends to stabilize over time. And despite the fact there's a continuing decrease in, say, in MMSE scores. So these are available. What we've been working on is something called cell-free DNA with Zach Chatterton, who was in Sydney and now moved to Melbourne at the beginning of the year. So cell-free DNA is interesting because it's related to this extracellular DNA that you find that is free-floating within the blood. And this is a technique that has been used to detect fatal genetic abnormalities. So we can take a little bit of blood from the mom and then test for various disorders, because you will find in blood so cell-free DNA from the mom and from the fetus. It has been used also in cancer to diagnose and track the progression or remission of various kinds of cancers. So the idea here was to use this knowledge and test whether there are some markers using cell-free DNA that would be indicative of disease in frontotemporal dementia. The difficulty is that you're talking about the same body, so there's cell-free DNA from a single host. So as opposed to like here you have a DNA from the baby and from the mom, or here you have DNA from the cancer compared to the non-cancerous cells. But what Zach did was to use the epigenetic, the methylation aspects of DNA that can identify the different types of cells where these cell-free DNA come from. So using that, what we found with taking cases of behavioral variant frontotemporal dementia, there was correlations between cortical thickness and anteriorly, so dorsolateral prefrontal cortex on the left and the right, and anterior cingulate and mid-cingulate between the cortical thickness and the amount of cell-free DNA that we found in the blood. And in particular, there was a range of regions that were identified that were all related to regions that clinically make sense. We know that we've seen changes in cortical thickness. So right insula, middle temporal, on left and the right, frontal lobe cortex, superior frontal cortex. And what you can see is that the relationship between the amount of cell-free DNA, fragments of cell-free DNA that you find in the blood in the cases, in patients, but there's no associations with controls. Obviously, we're talking small numbers here, about 15 cases versus 10 controls, but it seems to indicate that there's something that can be done using this technique. And particularly in relation to that really have raised our interest is the relationship in the insula and the superior temporal. Because we know that the insula is related to, or atrophy in the insula is related to emotional processing and interoceptive integration of information. And then the superior temporal lobe in the left hemisphere is associated with some aspects of language processing. So there's some potential use in terms of role for regions of interest in clinical trials that are outcome variables, outcome measures to look at changes in the amount of atrophy or cell-free DNA related to these regions. Now, I'm mindful of the time. So I'm going to skip through some of these slides, because the other aspect that we've been working on trying to resolve some of these known unknowns is to find ways to measure interoception as a cause of or underlying causes for some of the disabling clinical features that occur in these frontotemporal dementia syndromes, and in particular emotion processing, because emotion processing is related to the capacity to, well, decipher social cues and maintain interpersonal relationship, which is obviously a main component of who we are as humans. So that's work that has been done by Jessica Hazelton as part of a PhD. But essentially, what we found, and I'm going to skip through that, it's a multi-center study that we did with collaborators in South America. But it was a nice study because it was very simple. It was an interoceptive task where patients were either asked to press a button whenever they heard a heartbeat played through a loudspeaker, so the external aspects, which we call exteroception. And they had to do the same thing, but whenever they felt their heartbeat within themselves. And then what you do is you compute a mean distance index. So that measures the discrepancy between the actual heartbeat or the sound and the button press by the patient. And you do that by doing simultaneous ECG recording. And then we correlated that with capacity to recognize emotions. And building models, what we found first was that an emotion recognition task, all patient groups are impaired compared to controls, which is not surprising. In the exteroception, all groups are preserved. So the capacity to press a button in relation to an external sound is intact. But when it comes to monitoring your own internal heartbeats, then the only group that was impaired was the behavioral brain FTD patients. Then we built a model trying to understand which variables would be predictive of emotion recognition performance. And essentially, you end up with a model that has 4 main variables. One is the cognition. So the more impaired you are, the more difficult it will be to complete the task. But then there is only two other aspects. One interaction terms between cognition and the diagnosis for Alzheimer's disease and Parkinson's disease. So the combination of having a diagnosis, so it means that the more impaired with the diagnosis of Alzheimer's disease will impact more strongly on your emotion recognition performance. Whereas in behavioral brain FTD, this is not the case. So cognition does not play such a strong role, but it's more the interactions with interoception. So if you have a diagnosis of behavioral brain FTD so the interoceptive deficits associated with these diagnoses will impact more strongly on your capacity to recognize emotion. So there we can differentiate very different mechanisms in terms of emotion recognition performance. And we also did some neuroimaging, which I don't have time to go through. But just to summarize, so again, there are some known knowns that we are better at knowing. So there's certainly some improvement in our understanding of the characteristics of these different syndromes. And also, but the challenge is still to understand this overlap, particularly with disease severity. When it comes to the known unknowns, the prediction of disease progression for individual cases. So these individual management remain a significant challenge. And at this point in time, it's still, I think that we'll make progress with these biological markers. It's still early days in that, but there's progress to be made there. Neuroimaging is certainly useful to confirm the diagnosis, but less so in identifying pathology. And finally, in vivo biomarkers work, as I said, it's still early days, but it's really promising findings that certainly will help with drug trials in the coming years. I'm going to stop here. All this work is done by a large group of people. And over the years, we've had numerous talented people working on these different aspects. And that's the last lab photo pre-COVID, so it's dated a bit, but I just acknowledge the contribution of all these people and also the funding bodies that have been supporting our work. So from the Australian government, the RC, and the National Health and Medical Research Council. And thank you for your time.
Thanks, Olivier. That was quite the tour through all things frontotemporal dementia. You covered a lot, so thank you. We have some time for Q&A, which is great. So if you have any questions, please submit them now through the chat function. Alternatively, if you'd like to ask your question to Olivier in person, turn on your camera and microphone and we can call on you in turn. So while we're waiting for people to either get their typing on or switch on their camera, I have a question about, I mean, you covered so much, but just a review of your thoughts on what's happening in the clinical trials area, especially in terms of disease modifying treatments. What are the agents that are kind of in the pipeline, Olivier?
Yeah, look, in FTD, there's not much. At the moment, the main clinical trials are targeting the familial forms of FTD, because again, we know the pathology. So there we are involved in 3 or 4 clinical trials, primarily targeting granulin mutation carriers. So the pre-symptomatic and symptomatic mutation carriers. One is a phase 1, which is a single injection of something in the back of the brain. So it's a one-off injection. And then it's follow-up over 5 years. And the main outcome is to see whether there will be an increase in the level of granulin in the brain over time, and also a delay to disease onset. Then there's a phase 3 clinical trial in granulin mutation carriers that are for symptomatic mutation carriers, so people showing signs of the disease already, as well as family members in the pre-symptomatic phase.
And are they mostly kind of monoclonal antibodies and selenate, or is there anything else going on?
Well, yeah, then there's selenate, but selenate is targeting tau rather than, and it's in sporadic cases, and it's targeting tau rather than TDP. Then there's a trial that we stopped, but it's more at the symptomatic level. So it's a symptom modifying clinical trial using oxytocin and for behavior and trying to improve these social aspects of the behavior.
That's really interesting.
But yeah, the problem again is to target or to manage to recruit patients with the right pathology, because obviously if you have something that is specific to tau and you have in your clinical group patients with underlying FUS or TDP-43, then that's going to be a problem.
We have a question from Chiara Bansal in the chat. Which is more common, behavioural variant or language variant FTD? And are there ethnic, gender or age risk factors for each subtype?
Yes, the behavioural variant is the most common subtype and probably accounts for 62/3 of cases of frontotemporal dementia. Then the difficulty is always, categorization. I was thinking about that, about something else, is that, I don't know if you remember, I went through these very quickly, but the Murley paper was presenting these nice little boxes of different syndromes. But really, when in your clinic, it's rare that you will find someone who tick all the boxes, the right boxes, and just for that particular syndrome. So it's very more common to see someone with a little bit of behavioral changes, as well as a little bit of language problems. And then when it comes to which box are you going to tick, it's more which is more prominent at that point in time. So sometimes you will tick the box behavioral variant FTD when there's still some semantic loss associated with it, but probably not to the same degree. So it's still very difficult to determine the exact prevalences of these different syndromes. Second part of the question, risk factors, ethnic differences, that's a very good question. I don't know, because the way we're a clinical research group, so we're very limited in the number of patients that we can see. So our inclusion criteria are quite restrictive in the sense that we need to see people who have a reasonable level of English to participate in our studies. So it means that, and it's an acknowledged limitation of the work that we do, is that people from non-English speaking background, we don't have as much information on them. So we've started doing some work with our collaborators in South America to try to understand, particularly in the language presentations, whether there are some similarities in terms of the unfolding of the clinical profiles and change over time. With regard to age, yes. As I said at the beginning, there's really a peak of onset in the sporadic cases in the late 50s, early 60s. But you still have cases who will present in the clinic in the early 40s to the late 70s. A younger age tends to be associated with a familial form of the disease, particularly in the tau mutation carriers.
Great, thank you. That was very comprehensive. If everyone could join me in thanking Professor Olivier with your clap reactions. I think we've all learned a lot about FTD, so thank you. So next fortnight we have Dr. Helen Barry present on using citizen science to explore the age friendliness of cities. So please join us then. You'll receive the registration link and the information by email. So please join us next fortnight on Tuesday, the 11th of October at 12 p.m. And thank you for attending our seminars in Aging program.